Hello everyone. So far we have been discussing the fundamentals of nuclear chemistry and then some of the analytical techniques based on nuclear phenomena and also the radiochemical techniques. Today's lecture I will describe some of the applications of radioisotopes in societal applications in the service of mankind, like healthcare, industry, and so on. So before going to the actual applications, let me explain what are the fundamental principles underlying these radioisotope applications. So there are mainly three concepts or three properties of these radiations that enable them to be useful in many fields. So I'll just explain them. The first one is the radio tracer concept. I had also explained it earlier, one of the lectures on radio tracer based analytical techniques. And the underlying principle is that the radioisotopes have the same chemical properties as their stable isotopes. So this property of radioisotopes helps them to trace the path of their stable isotopes. You take any industry or even a chemical reaction where you want to understand the mechanism of the chemical reaction, then you can label one of the atoms with the radioactive atoms and see how this particular reagent is moving in the chemical system. Like sulfur, sulfur containing chemicals, their reactions can be studied by sulfur 35 labeled compounds. In fact, many of the reaction mechanisms have been understood using radio tracers, isotope labeled compounds. So we will discuss that applications in many areas. Second property of these radiations is that these radioisotopes emit radiations that are highly penetrating, and because of that high penetration power, you will find many applications of radioisotopes in industry like radiography or even in the non-destructive testing investigations. You will see them, many of them. So this is the main concept. They can penetrate through thick absorbers. Like what I have shown here is that alpha particles will travel this way. Like if we have do a transmission experiment, then I by I0 will follow 4 alpha particles. They will travel same distance, and after that there will be certain fall. That is the typical transmission graph for alpha particles, whereas the gamma radiation will follow exponential decay. So the like Beer-Lambert law. So you can make use of this intensity decrease with thickness of absorber in many applications. And the third is the effects of radiation. Now, ionic radiation can create defects. They can generate free radicals. They can produce heat. They can even kill pathogens, and they can damage the cells. And these properties of these radiations find them very useful in medicine, for example, in cancer treatment, therapy of cancer, and also modifying materials by polymerization, or you can create defects in materials like diamonds. You can create the different colors in diamonds and add their value. They produce heat. You can use them as a source of heat, like 238 plutonium is used in satellites as a source of heat. You can power like pacemakers. Then they can kill pathogens, so that this is utilized in food technology. You can prevent the sprouting of the fruits and many applications. They can even induce mutations in seeds, and you can produce improved varieties of crops by using radiation-induced mutations. So the list is endless, and I will cover some of the examples of these three concepts in my talk. Okay, so first let me, in this particular talk, discuss the applications of radioisotopes in healthcare. And these applications could be based on radio tracer concept, or they could be based on the radiation effects on in the human body. So all sort of applications can come. So in radio isotope applications, healthcare, one of them is in vitro. That means in the test tube, in the laboratory, you can do analytical determinations of the different biological components of our human blood or many other biofluids you can determine. For example, we discussed in the radioanalytical techniques the radio immunoassay, where you can determine the concentration of T3, T4, TSH, or even other like prostate specific antigens you can determine by using labeled antigens and interacting them with antibodies, and then finding out the concentration of these molecules at very low levels like nanomolar concentrations. But they are done in the laboratory. In vivo, in the body, if you introduce radioisotopes, then you can do investigations of diseases, and the chemicals that are introduced in the body in vivo, they are called radiopharmaceuticals. They are like medicines, they are pharmaceuticals compounds, but they are labeled with the radioisotopes. So we'll discuss them in this lecture. And also the radioisotopes, by virtue of their radiation effects, by virtue of killing the tumor cells, we can use them in therapy. And lastly, the sterilization of medical products. These medical products, when they are implements, the tools, the scissors, the tweezers, and different implements used in operations. So they have a chance of getting, infected again during handling. So in this particular case, what happens? You can pack them in a cardboard boxes, take them to the plant for irradiation, and after irradiation, they will be opened only when they are being used for the operation. So you can sterilize the medical products, implements, tools by using radiation. So I will not discuss this much in this particular talk, but I will talk about other first two, diagnosis and the therapy. Okay, so now let us discuss what are radiopharmaceuticals. So radiopharmaceuticals are pharmaceutical compounds, but they are labeled with radioisotopes. So a special radiochemical formulation, the formulation is a radiochemical, it's a compound of adequate purity. So it should have a desired radiochemical purity, radionuclidic purity, that is what we mean by adequate purity, and pharmaceutical safety. So there are pharmacopoeia for different quality control measures to be taken with regard to its pH, sterility, and so on, pyrogenicity. So it should be suitable for oral or intravenous administration to humans for performing a diagnostic test or for treatment. So any compound to be injected intravenously or orally in the human body has to pass the tests of this kind of quality control measures. So the pharmaceutical safety is important, so that we will discuss in my talk. So this is a general definition of a radiopharmaceutical. Now, the radiopharmaceutical can be used in diagnosis of diseases, wherein we take the image of an organ. It could be static, or we can see the dynamic flow of the body fluid, the blood. Then we can get the dynamic image of an organ, how the metabolism is taking place in the human organ. So that you can do using diagnostic applications, where we inject the radioisotope labeled compound, and then in the body, wherever this radioisotope is going, that particular organ we can do. They take the image in static or in a dynamic, or you can have sequential images. So this is another application. And then we have the therapeutic applications, where you can use radionuclide externally. It's called teletherapy. From outside, you can expose the cancerous tissues and kill them, or we can introduce the radioisotope in the body either by a sealed source in a cavity or by using a radioisotope labeled compounds. So these radioisotopes essentially are emitting those which are introduced in the human body in the form of a compound, like radiopharmaceuticals. They have to have particulate emission like beta plus or alpha, beta minus or alpha, so that they can do the damage to the cancerous tissues. So in radiopharmaceuticals, if you inject into the human body, then they are required to have particulate emission beta, alpha, so that they can kill the tissues in their nearby vicinity. So that is the kind of requirement we have for radiopharmaceutical in therapy. Okay. So let us see for diagnosis diagnostic applications, what are the kinds of radioisotopes we can choose for diagnosis as a radiopharmaceutical? So one of them is the decay mode. The decay mode, see what we are doing, we are going to take the image of the organ in which the the radioisotope is distributed. So it should preferably emit only gamma ray. So it should not be emitting a particulate, because that particulate will unnecessarily damage the tissue. So there are not many radioisotopes which will be not emitting particulate, because the gamma is emitted only after beta or alpha decay. So, but there are like internal transitions. The isomeric state is there, like technetium-99M. It will decay by only internal transition. 140 keV is a gamma ray and having half-life of 6 hours. That is an ideal for diagnostic radiopharmaceutical. First is that it should not emit preferably any particulate emission. The second is that the energy of the gamma ray should be in the range of 100 to 200 keV. Why? Because if it is below the 100 keV energy, then it will be stopped in the body itself. It will not come out of the body. And if it is more than 200 keV, then the efficiency of detection will be low. So 100 to 200 keV is the optimum range of gamma energy for a good image. That is the reason why gamma energy should be in 100 to 200 keV. More than 200, efficiency will go down. Less than 100, the gamma ray will not come out of the body. It will be attenuated by the human body. Half-life should be short, preferably few hours. Again, the reason why it is so, because you don't want the radioisotope to be in the body after the image has been taken. So it should come out of the body, or it should die down in the body itself. So that few hours time is optimum for the diagnostic investigation. And lastly, it should have a versatile chemistry. That means it should be able to complex different ligands, and it should, those ligands having high affinity to different organs. So a particular metal ion in much different oxygen state will be ideal, because different ligands may bind a metal ion in different oxygen states. So some of the examples of these radioisotopes that are used in diagnosis are technetium-99M, ideal. In fact, it is called the workhorse of nuclear medicine. About 80% of the nuclear medicine investigations for diagnosis are done using technetium-99M. So gamma ray is ideal, half-life is ideal. Indium-111, ideal gamma ray energy, but half-life is little long. 123 iodine, half-life is and gamma ray wise ideal. That is why there is huge demand for 123 iodine, though it requires a xenon target, enriched xenon. Thallium-201, you can see half-life and gamma energies are quite good. Gallium-67, 3.26 days and good. And iodine-131, though the half-life is long, you will find in many applications it is very, very useful, because it can be used in both diagnosis and therapy. It has the gamma ray energy, it has the particulate emission also. Now, once you label a compound and prepare the radiopharmaceutical, there are certain pharmaceutical quality control measures you must establish, and this they belong to the physicochemical control, radiochemical control, and biological control. In physicochemical properties, you should inspect the solution, that it should be clear, there should not be any turbidity. pH should be the proper zone, which will be compatible with the body. The chemical purity of the compound, there should not be any other impurities. And sometimes you use in the particulate form, some microspheres are used for some treatment, so the particle size should be in the proper range. The radiochemical control with regards to radioactive concentration, what is the concentration of radioisotope, is millicurie or microcurie or curie. So, depending upon the requirement specified by the doctor nuclear medicine personnel, you should specify that concentration. Radionuclidic purity, there should be no unwanted radioisotopes, and the radiochemical purity, the chemical form of that molecule should be whatever is the desired one. For example, if you have fluorine in fluorine FDG, then FDG should be only, there should not be any other derivative of FDG. So that kind of purity has to be established. So there are measures to specify to determine the radiochemical purity by like thin layer chromatography or pipette chromatography, radionuclide purity by gamma spectrometry, radioactive concentration by counting the sample. Then you have the biological control, sterility for injectables. You have to have it should be sterile. It should be the pyrogenicity means it should not produce heat in the body. So this is an important requirement of the pharmaceutical, and it should be distributing in the organ of your interest. So these are the properties that the pharmaceutical should have. Okay, so I've just given you a figure that whatever different organs in the body which are likely to get affected by a disease, then there are radiopharmaceuticals, both in terms of the radioisotopes as well as the molecule, for which there has been already there in development to produce those kind of pharmaceuticals which will be selectively going to the particular organ. For example, if you want to see the glucose metabolism in the brain, then you have fluorine 18 labeled FDG. If you want to see the thyroid uptake, thyroid functioning, if whether there is any malfunctioning of thyroid, you can do sodium iodide labeled with 131 iodine, or even you can do technetium 99M, because technetium also can go to thyroid. Then for heart, you have F18 FDG, and there is one thallium also, thallium 201 also will go to heart, so you can do for stress test. Then for bones, you have ethylene diamine tetramethylene phosphinic acid, sulfur samarium labeled EDTMP, or you can have phosphorus labeled orthophosphate. Again, for the bones, you have holmium 166 hydroxyapatite. So, in fact, there are some of the few. You will find that, for example, for the prostate, there are prostate-specific antigens. So, if you label that antigen with a particular compound like lutetium, it will selectively go to prostate, and you can do the image as well as you can do the treatment of the prostate cancer. So, for every organ, almost every organ in the human body, you will find there is a particular pharmaceutical, and there is a proper labeling radioisotope. So, you can do imaging of different organs of the body. Now, what are the experimental techniques for this? You have this technique called for imaging scintigraphy, and it is also called the single photon emission computed tomography, SPECT. So the principle of SPECT is that suppose you have the organ which is containing the radioisotope here, and this organ is now in the human body, it is emitting gamma radiations isotopically. So you from outside, this is the machine for SPECT. So the camera, this is the camera. The patient is made to lie on this, and the camera will detect the gamma ray coming out from the organ. Now the radioisotopes are emitting gamma ray in all directions, but you have detected only one side, and you want to take the spatial image. So as a function of in space, how the organ is distributed, that means how the activity is distributed in the organ. So you have a detector where the gamma ray is made to go to the detector through collimators. So it will go at a particular angle, so that tell you in which bin this gamma ray has come. So this is a segmented kind of system. So this collimation helps you finding out the place from which the gamma ray has come. Then the gamma rays are detected by the detector, mostly scintillated sodium iodide, thallium, or BGO type of scintillators. And there are PMT photomultiplier tubes to convert the signal, the photons converted into electrons at a PMT, and then there is a position sensitive circuit to identify from which place in the organ the particular gamma ray has come. Like for example, this collimation will give you this particular position. So we can see in organ, and you can construct the image by help of a computer. So that is the principle behind the SPECT. So it essentially gives you the 3D image of the organ in static or in dynamic. Like this is just to give an example, SPECT images like for this technetium 99M labeled MIB, it is called MIB, methoxyisobutyl isonitrile, and it is a heart imaging agent. You inject intravenously this technetium 99 MIB, and after 15 minutes you take the image of the heart of the person from outside. Externally, you are non-invasively, you are monitoring the gamma rays that is coming out of the human body, and you can see as a function of time how when the heart is pumping the blood, you can see the different images, different cross sections of the heart, and if a particular portion has got infructuous, that means if the blood is not reaching there, the muscles are dead. You can identify. The doctor can identify which portion of the heart has become infructuous, means it is dead. So that kind of a investigation can be done using radiopharmaceuticals labeled with proper radioisotopes. Similarly, you have this scintigraphy image of lutetium-177 labeled EDTMP in dogs, which were found to be having osteosarcoma. So you can see this is how you keep the patient in the SPECT machine, and you can see the inflammation. So that sarcoma, that the particular tumor in the animal can be seen in bright intensity of the radiation. The radioisotope is going and depositing there. So you can see that you can, even the lutetium-177 not only can do the diagnosis by means of gamma ray, you can also do the treatment, because the therapeutic application requires emission of high energy beta particles, which lutetium-177 emits. Now that was the SPECT imaging. So you use a single photon emitting radioisotope and you take the camera and it will take the image of the organ. But the resolutions in the SPECT are not very high, and therefore the positron emission tomography PET has become now more popular because of the resolution in terms of the space. The spatial resolution is excellent in PET. And PET, you require a positron emitter. The positron emitters are carbon 11, nitrogen 13, oxygen 15, fluorine 18, and also gallium 68. You can see their half-lives are minutes, and the beta plus positron energy and the way they are produced. So F18 is in fact these days most commonly used because it is having a half-life of about 2 hours. So from the cyclotron, they can be transported to the hospitals. Whereas the other isotopes, carbon 11, nitrogen 13, oxygen 15, their half-lives are even less than 20 minutes. And so they require the cyclotron to be present in the hospital. So immediately you transport the pharmaceutical to the hospital within few minutes, and then you can do the PET analysis. But fluorine 18 you can take from cyclotron to other hospitals in the city. And another thing is gallium 68 is another very interesting pair. It's a generator. You have germanium 68 decaying to gallium 68, and you can take the generator to the hospital, and we milk the gallium 68, which is then injected into the human body for PET investigations. So this is again the principle behind the positron emission tomography. So now you have a ring of detectors. It is not single detector. You have a ring, and the patient, like MRI, it is like MRI. So the patient is made to lie here. The detectors encircle the patient. So suppose you have an organ here. It is odd shaped organ, and then from different parts of this organ, the gamma rays, the positrons are anyway annihilated, so gamma rays are emitted, and the 511 keV gamma ray, they are emitted at 180 degree. So you know the positron annihilation, and so from a particular point you will see that the two detectors will receive an event at 180 degree. So similarly, this event is these two events are coming from this place. So this, because of the coincidence phenomena, you can identify from which place in the organ the 500 keV gamma ray has come. So this is the schematic. You have this is actually an annular one. This is a circle. It's a cylinder type of thing, and then this is the BGO detector, and it's like a circular array. And then this data are fed to the computer to do the investigation, to compute, to simulate the distribution of activity in the organ. So for a PET machine, it's a very compact machine, and in the cyclotron, once you produce the positron emitter, you have to have the very fast chemistry. There is a radiochemical unit which will separate the radiochemical, and then you can label them and then transport to the hospital. So this is the general principle of the PET machine. And the images that you get from the SPECT versus PET, you can see this is the image of a brain using SPECT and PET. So you can see the marked difference, improvement in the image of the brain using PET vis-a-vis the SPECT image. That is why now PET is becoming very, very popular among the nuclear medicine practitioners. Now let me come to another application of radioisotopes is in therapy of cancer. And here there are three techniques. One of them is teletherapy. As the name itself implies, the radioisotope is from outside the body, and you focus the beam of the gamma ray onto the organ that is to be irradiated. People use cobalt-60 source, which is emitting high energy gamma ray. 1.17 and 1.33 MeV. And the gamma rays are focused onto the tumor, so you have to put it in the proper shield and then allow only a beam of gamma ray to come, though the gamma ray is emitted in all directions, but you are collimating the gamma ray to come to a particular tumor. So there is a huge activity required, because you cannot focus the gamma ray, only you can collimate. So about 12,000 curie of cobalt-60 is used in one machine for this teletherapy. So this is the where the patient lies, and this is the machine, the source cobalt. So you can, it is in that is sealed, and the gamma ray emitted is the cobalt-60 is emitting gamma ray to irradiate the particular organ. So though cobalt-60 is most commonly used, there are now advancements going on. You can produce gamma ray from electron beam by means of Bremsstrahlung. You can use protons for therapy, and you can use boron neutron capture therapy. That means, suppose you want to produce protons, you can have neutrons. You can have a neutron source for treatment of the cancer. So you use borons, and the neutron will induce the reaction in the boron and give you alpha and lithium-7. So this alpha and lithium-7, essentially what will happen? So this alpha and lithium-7 are now highly charged particles, and they will damage the cancerous tissue. So you have to take boron-labeled compound with that organ, nearby that organ, and bombard with neutrons selectively, because boron has very high cross-section for neutrons. You can do the damage of cancerous cells by using this technique. So there are many techniques which are being used for cancer treatment. One of them is teletherapy, other one is brachytherapy. Not all cancers can be treated by teletherapy, because from outside you cannot take the gamma ray to a particular organ, particularly the deep-seated tumors like cervical cancers, prostate cancers, ocular cancers, eye cancers. So these are deep-seated, so you may unnecessarily damage the other organs. So those tumors which are not amenable by teletherapy, they are treated by brachytherapy. In brachytherapy, you use tiny sources. They are placed in a cavity, which can be placed in a cavity, and particularly people use 192 iridium and 125 iodine. 125 iodine has a half-life of 60 days, and gamma ray energy is 35 keV or so, and 192 about 100 200 keV gamma rays. So you put the sources for a particular period of time, and after that, these are sealed sources, so they are not going in the bloodstream. You can just take them out after proper exposure. These are the typical, this needle-shaped pins source. They are sealed sources. They are called ocuprostra sheets because they can be used for ocular cancer as well as prostate cancer. So they are made in the laboratories, and this is the typical drawing of the seed. You can see the length of this is 4.5 millimeter, and the diameter is of the 0.8 mm. So very thin, tiny needles and wires, like wires. So they can be placed behind the eye, and can placed in the other places of the human body. So wherever you want to do the treatment, the prostate cancer, so you want to irradiate the prostate with this gamma rays, you can do that. And then lastly, we have the radionuclide therapy. Radionuclide therapy is that, like radiopharmaceuticals used for diagnosis, that you can tag the radiopharmaceutical with a radioisotope which is emitting the particulate emission, and then do therapy. So the pharmaceutical will take the radioisotope to a particular organ, and the radioisotope will emit the particulate radiation alpha or beta, and that will kill the cancerous cell. So 131 iodine is used for thyroid disorders, yttrium 90 for treatment of arthritis, and phosphorus 32 for skin cancer. Yttrium 90 and phosphorus 32 are pure beta emitters. The high energy beta is destroyed the tumor cells, and so for example, iodine, you can use the beta for thyroid disorders. So even for therapeutic agents, if you have some joint diseases like arthritis, then there are radioisotopes like yttrium 90, erbium 169, rhenium 186. They can be used for treatment of joint diseases. One of the examples you can see here, immediately after the treatment, the distribution of you have the arthritis, so there is some fluid deposition, and that you can take the image of that knee with these radioisotopes. after 3 months of treatment or after 6 months of treatment, you can see that the patient is recovering, that particular inflammation is going off. So this image is taken with yttrium-90 hydroxyapatite for treatment of arthritis. So we can use these radioisotopes which are emitting particulate radiation for treatment of many types of diseases. And lastly, there are theranostics, means therapy and diagnosis. So they have dual purpose of not only doing therapy but also diagnosis. So they are called, this is a new concept coming up last few years now, the radioisotopes having dual properties. So they emit particulate radiation like beta alpha, and also they emit gamma radiation, and therefore they can be used for both diagnosis and therapy. So for example, when a patient is undergoing iodine therapy, you can monitor the therapy, what is happening to the patient as a function of time using gamma ray emission. So iodine 131 is one of the isotopes. It has got a beta minus and a gamma ray. And 177 lutetium is another isotope which has got a very good chemistry, ideal gamma ray energy 100 to 200 and half-life and particulate radiation. So this is the image of lutetium 177 hydroxyapatite, which is used for treatment of arthritis in medium-sized joints like elbow, wrist, and ankle. And you can see immediately after therapy and then after 1 month of the therapy, how the inflammation is vanishing as a function of time. So this is how one can carry out investigations of therapeutic applications of radioisotopes. You can do teletherapy, you can do brachytherapy, and suppose there are the tumor is in the glands, sometimes even the brachytherapy will not help. So you can go for radionuclide therapy, and the theranostic ones help you not only in therapy but also in diagnosis as a function of time while the therapeutic treatment is going on. So that's all I had to say in this particular one. The next lecture I will talk about the other applications of radioisotopes in industry and so on. Thank you very much.